Hi, my name is Jason Lee, and these are my partners David Liu and Alan Lee. Our project was the identification of compounds to overcome carbapenemase-related multidrug resistance in Enterobacteriaceae. Multidrug-resistant bacteria are rapidly becoming a major worldwide concern. President Barack Obama issued a White House executive order to combat antibiotic-resistant bacteria earlier this year, and Britain's Prime Minister, David Cameron, released a statement stating that if we fail to act, treatable infections and injuries will kill once again. But how big of a problem are multidrug-resistant bacteria? Well, here we use Enterobacteriaceae as an example of multidrug resistance. According to the Center for Disease Control, 140,000 Americans contract Enterobacteriaceae infections every year, 26,000 of which are multidrug resistant, leading to around 1,700 deaths and an additional $40,000 in medical expenses per infection. This is an enormous problem. The production of extended spectrum beta-lactamases is the most common mechanism of drug resistance in bacteria, and it is capable in activating virtually all known beta-lactam antibiotics. So beta-lactams are typically characterized by a beta-lactam ring, as shown by this red circle over here. They are the most commonly used group of broad-spectrum antibiotics and inactivate the penicillin binding protein and therefore disrupt the bacterial cell wall synthesis. Here are three types of beta-lactams: penicillin, cephalosporins, and carbapenems. We focused on carbapenems because they are considered last resort medication and typically used in life-threatening infections. So here we show the base structure of a carbapenem, marked by its beta-lactam ring. Carbapenemases bind to the carbapenem, hydrolyze and cleave the beta-lactam ring, and inactivate the carbapenem. The two most common types of carbapenemases are Klebsiella pneumonia carbapenemase, also known as KPC2, and the New Delhi metallo-beta-lactamase, also known as NDM1. Both are commonly found throughout the world and facilitate resistance to most beta-lactam antibiotics. So we analyzed these two carbapenemases and first compared their primary sequences using Clustal. In this analysis, asterisks represent identical amino acids, while colons represent similar amino acids. We found that there is very little homology between the two. Next, we compared the tertiary structures of the two carbapenemases, and again, we saw a low degree of homology. So how do these two proteins that are so different in structure both exhibit carbapenemase activity? Well, to answer this question, we took 10 different carbapenems and docked them into the structures of KPC2 and NDM1 using Autodock Vina. We found that the predicted binding affinities of each carbapenem between the two proteins were quite similar, as shown in this table. This suggests that the active sites of KPC2 and NDM1 are also similar, so we may be able to find a compound that inhibits their activities.
Our goal is to discover potent small molecule inhibitors for KPC2 and NDM1 that can synergize with and restore the carbapenem's bactericidal activity. We hypothesize that taking a computer-based approach will facilitate the identification of novel inhibitors for KPC2 and NDM1. This method has been used in the past to target other beta-lactamases. This diagram here shows the three different approaches we took with virtual screening to identify potential carbapenemase inhibitors. These three approaches will be explained in detail later. All candidate inhibitors that were identified through virtual screening were subject to biological testing according to this diagram. We used Klebsiella pneumoniae strains, which are known to express KPC2 or NDM1, and confirmed that they were resistant to imipenem at 1 millimole. We then mixed the Klebsiella with 1 millimole of imipenem in combination with various concentrations of the potential inhibitors and incubated them for 2 hours. Afterwards, we monitored the bacterial growth with microplate readers in triplicates. Now, this assay can have three potential results. First, there would be no inhibition compared with bacteria alone. This indicates that the compound has no activity. Second, there could have been inhibition of bacterial growth regardless of imipenem. This suggests that there was nonspecific inhibition by the compound through unclear mechanisms. Third, the bactericidal activity of imipenem would be enhanced. This indicates that the compound may be a true carbapenemase inhibitor.
So the first approach that we took to identify inhibitors for both KPC2 and NDM1 was a fragment-based approach, utilizing DocBlaster, an online virtual screening server. So what we did was we uploaded KPC2 and ND1's PDB structure to the web server, and then we submitted a job which docked the 1.5 million compounds of the zinc fragment-like subset into the active site of each enzyme and then calculated its binding affinity. Doc Blaster listed the top 200 compounds, 30 of which we purchased based on both their rank and availability, and then tested them in an experimental assay. Here are 5 candidate compounds along with their 2D molecular structures, predicted binding affinities, and IC50s against bacterial growth. At the bottom of the slides are bar graphs showing the bactericidal activity of each compound. The light blue bars represent the bactericidal activity of the compound by itself, while the navy blue bars represent the bactericidal activity of the compound in combination with imipenem. 3 compounds seem to have activity against bacterial growth, with IC50s of 1 millimole and 1.8 millimole against KPC2, and 0.5 millimole against NDM1. However, when you look at the growth curves for compound O is the classic example of a nonspecific inhibitor, with relatively similar bactericidal activity with and without imipenem. Compounds P and M, on the other hand, seem to be more promising carbapenemase inhibitors, since they have a notably higher bactericidal activity when combined with imipenem. So we will further test whether a compound is a true carbapenemase inhibitor or just a nonspecific inhibitor by conducting another biological assay, which will be used to calculate the combination index of each compound. This will be discussed further later. So the second approach that we took to identify inhibitors for KPC2 specifically was a structure-based approach, utilizing iStar-IDoc as a virtual screening server. So what we did was we uploaded KPC2's PDB structure to the web server, but before we actually submit a job, we first used iStar-IDoc's ligand filtering tool to filter out all compounds that did not comply with Lipinski's Rule 5. We then submitted a job and took the top 100 compounds as identified by iStar-IDoc and further filtered them using RDoc Vina and Pyrex and IGEMDoc, which predicted that only 6 of these compounds would have a better binding affinity to KPC2 than imipenem has. We purchased these 6 compounds and tested them in an experimental assay. Here are the 6 compounds along with their predicted binding affinities and IC50s against bacterial growth. Notice how all 6 compounds have a better predicted binding affinity to KPC2 than imipenem has. Using the same biological assay as described earlier, 2 compounds, D and F, had activities against KPC2 with IC50s of around 1 millimole. Here are the doc poses and protein-ligand interaction diagrams of the 2 compounds as predicted by iStar-IDoc and plotted by PostView. Interestingly, iStar-IDoc predicted that both of these compounds bound to at least 2 out of the 3 conserved crucial active site residues, which in this case were serine-130, threonine-235, and threonine-237. This may explain their better activity against KPC2 when compared to the other compounds tested, which were only predicted to bind to 1 or none of the active site residues. The third and final approach that we took to identify inhibitors for NDM1 specifically was the ligand-based approach, also known as pharmacophore modeling. So dimercaprol was an already identified NDM1 inhibitor when the IC50 of 10.4 micromoles against its purified enzyme. So what we did was we uploaded dimercaprol's 3D molecular structure to ZincPharmer, an online pharmacophore search software, which identified 4,000 compounds that had similar pharmacophores to that of dimercaprol. We then further screened these compounds using RDoc Vina, Pyrex, and IGEMDoc, which predicted that only 4 of these compounds would have a better binding affinity to NDM1 than imipenem has. We purchased these 4 compounds and tested them in an experimental assay. Here are the 4 compounds along with their predicted binding affinities and IC50s in combination with imipenem. One compound, compound J, had activity against NDM1 with an IC50 of 1 millimole, while captopril, a known NDM1 inhibitor, had an IC50 of 1.5 millimole.
The top 5 compounds from biological testing were further analyzed with the second assay. We used the combination index to determine the synergism of each compound with imipenem against multi-resistant Klebsiella. We incubated each of these test compounds in a 1:1 molar ratio, as exemplified by the table to the right. We then used the percent of inhibition from each of these pairs to calculate the combination index value using the CompuSim program, which uses these 2 equations shown below. The calculated combination index values from effective dose 50% to 95% for each of the combinations is shown in this table here. A combination index of value of 1 indicates an additive effect, while values greater than 1 represent antagonism and values less than 1 indicates synergism. Thus, the lower the combination index value, the stronger the synergism. Compounds P and F had low combination index values close to 0.1. This is further illustrated in the fraction-affected combination index plots below, where the simulated curves of both these compounds are well below the additive line when fraction affected is greater than 0.25. This suggests that these two compounds are highly synergistic with imipenem, enhancing its bactericidal activity. In conclusion, we took 3 different approaches with virtual screening to identify potential inhibitors for carbapennamases KPC2 and NDM1. We tested roughly 50 compounds by measuring their ability to enhance the bactericidal activity of imipenem. There were 5 compounds with IC50s less than 2 millimole when combined with imipenem. We took these 5 compounds and further analyzed them with the combination index. From this analysis, there were 2 compounds, P and F, with low combination index values and are shown to be highly synergistic with imipenem. In the future, this information could be used for further structure-activity relationship analysis. Ultimately, our goal is for the further optimization and design of a better compound for drug development. We'd like to thank our mentor, Dave Durrant, and our laboratory director, Dr. Kuraja. We'd also like to thank Ray Lee, Samantha Cope, Dana Delano, and Denise Williams for other guidance we have received. We'd like to thank our family and friends, including Stephen Wang and Andrew Jin, for all their support they have offered us in this project. Most of all, we would like to thank the Siemens Foundation, George Washington University, and Discovery Education for hosting this event and having us here. Thank you.